Thank you very much, ladies and gentlemen. I want to tell you today about a fantastic project that we're now embarking on. It's about applying the latest genome science to saving our critically endangered lowland subtropical rainforest. It's all about genetics, and our partner, would you please stand up, is Maurizio Rossetto, who's one of the great and acknowledged one of the finest rainforest geneticists in Australia and a leading figure in the world. So Maurizio, we value your input. Maurizio and I have known each other for 25 years, and during that period we have both progressed. He's gone to the top of the scientific heap, and I've had a good role to play in restoring our rainforest. The problem we're trying to solve is the lack of genetic diversity in many species in the lowland subtropical rainforest. The solution we're applying to it is developing a seed plantation modelled on a macadamia plantation that will contain, for the 23 species in our first plantation, 20 individuals that we have selected across their range to have the optimal genetic diversity to cope with climate change, to avoid inbreeding, and cope with other threats like pathogens. Let me talk first about the lack of genetic diversity in the plantings. You've heard about the fact that we've planted 1.5 million trees in the big scrub. Well, that figure now is actually 2 million. And when those trees mature, and they're being planted now at the rate of about 100,000 a year, would you believe? That will increase the area of the forest, to Carla's delight, by some 60%. So that's a huge achievement in itself. But unfortunately there's one issue, and that is the genetic diversity of the species used in those plantings, and that's up to 180 species, is inadequate. And the reason is that the seed collectors would only go and collect seed from easily accessible trees on the roadside or on a friend's farm, and they tended to collect from the same 3 or 4 trees year after year. We got some advice from Julia Playford, who 20 years ago, when we got this advice from her, was a professor of genetics, or botany, specialising in plant genetics at UQ here. And she said in those days, you need to plant with seed from 10 unrelated maternal trees, and they should be local provenance. Well, we've moved on from there, as Maurizio will tell you. If I'd like him to tell us a little bit today if we've got time, but he's giving a much more expansive talk about his Restoring the New program on Wednesday afternoon. So we realised, and we had great concerns for the 20 years about the lack of genetic diversity in these planted species. And if you think of the effort put in by landholders to plant now 2 million trees, and if you casually value those at $10 or $12 a tree, that's $20 million of landholder investment. And the tragedy is that those plantings won't survive. Most of the species will inbreed, and there'll be a loss of species, and instead of the great revegetated areas we have now, and we've got 14 acres on our farm, they will lose species progressively. So we've been thinking about this for years and wondering what to do about it. And there was a young guy called Maximo Bottaro who, as part of his studies at Southern Cross, I think it was for his honours year, did some DNA work on 3 of the commonly planted species, and he confirmed our worst suspicions that the genetic diversity was very poor and these species were doomed to inbreed. So I asked him how he got his leaf samples out of remnants, and he said, well, he developed a potato gun, which was a length of 50mm plastic tube, and he put a 90mm tube in the back and put a piezoelectric spark generator, filled it up with ether, fired it up into the remnant, and down came the leaves. And so I suddenly had a eureka moment, and I thought, well, if we can't get seed from remnants, why don't we get cuttings from selected trees? And in my simple approach, I thought, well, we'll select 20 trees from remnants scattered across the range from, say, Grafton up to the Noosa hinterland, which is the range of low and subtropical rainforest. I thought, well, random selection from 20 remnants is better than nothing. But then I talked to Maurizio about it, and he said, oh, that's a brilliant idea, but I want to bring the latest genetic science to bear on this. And what I want us to do is to collect samples from 5 or 6 old-growth trees in 30 to 40 populations across the range of these particular species. And so this was a bit of an eye-opener for me, and I thought, what a fantastic idea that is. So we've been trying to get finance for it. Maurizio and I did a comprehensive budget. We needed about $500,000 in cash for the 20-odd species. He would contribute to $300,000 or $400,000 in in-kind work. We could get together about the same, well, we could get to, say, at least $150,000 in-kind work, and that would match the $500,000. Well, I wrote 3 unsuccessful grant applications because I think the bureaucrats found this project a bit novel, and of course it had long-term implications, long-term outcomes. So I have one more application pending, and if that doesn't work, well, I'm determined that we'll raise funding from our donors to raise the $350,000 or the $500,000 to get this project off the ground. It's too important to not do. I'd like to now turn to the remnants, and as you've heard, the remnants around here are tiny, most of them. They're scattered, and the individual species within those remnants are even more isolated because of the 300-odd tree species in low and subtropical rainforest. The greatest number in any remnant is 180, and the big Scrub Flora Reserve, which is the biggest remnant left, which is 170 hectares, there's only 160 species. So you can see the species are even more isolated than the remnants, and in many areas their populations are small. And the likelihood is that they are too small to breed successfully. There will be inbreeding, and I think Maurizio said there's already some evidence of that. And of course, in our project, because we're sampling 6 trees from each population, we will be able to get a bit of an insight, at least to some degree, as to what the genetic variation within the remnants are, and then we can look at that across the landscape, and he will do his magic stuff to pick us the 20 best trees for the plantings. But when the plantings, which are continuing, when these trees start to produce seed themselves, and that gets disseminated across the landscape, the quality of the gene flow across the landscape of these species, and eventually our vision is to get to 100 species, not just 23. These good genes will spread across the landscape. They will be more successful in breeding. They should then, as they get into the remnants, they should outcompete the naturally regenerating species which have not good enough genes to survive. So we've got a plantation site. Well, I should talk about the partners first of all. Maurizio is the key partner in the Royal Botanic Gardens, and he's just fantastic. Rob Koyman, who's many of you know of, and he's talking here today. He's one of Australia's, and I guess the world's leading rainforest field ecologists, and he and Maurizio produce research papers in huge numbers. So he's a great colleague of Maurizio's. He's been a great supporter of our Landcare group for 25 years. So he's doing the cutting collection. We thought we had to make a start on this, so the Big Scrub Foundation has undertaken to fund the $70,000 budget for that activity. It's the first step in this project because we can't hang around any longer. We've got to get going on this. And it's going to be about 8 years to get to fruition, till we start harvesting seed from this plantation. So one of our partners is a guy called Rex Harris and his son, and they are big macadamia growers. They are the most innovative macadamia growers, and they apply the principles of biological farming to maca production, and that's what we're going to do in the plantation. He will do the site preparation for us. He will help us design the plantation, and he will help with the planting, and he'll do the ongoing maintenance. Fortunately, his farm where he has the most amazing array of gear and runs all these great trials, is only a couple of kilometres away from the site. So the other key partner is Mark Dunphy in Firewall Rainforest Nursery. He is the biggest rainforest nursery operator in the Big Scrub, and I think probably in Queensland as well. He will do the striking of the cuttings and growing on the planting stock from the cuttings, which will form the planting stock for the plantation. So the benefits, I probably should read these out because otherwise I'll forget some. I don't like reading, but I'll do it anyway. First, obviously, it will increase or improve the sustainability of the plantations, and I'm hoping that the people that have already planted big areas will be able to plant additionally beside them. In my case, I think we'll probably knock out some of the areas we've already planted, and we'll replant some of these genetically better species. The other thing we need to do, which I should mention actually, a big shortcoming in many of the plantations is that the species are sort of distributed willy-nilly. And to get effective pollination in most rainforest species, which are insect pollinated, they need to be within 50 metres of each other, and certainly no more than 100. Whereas in most cases, the individuals of one particular species are just scattered across these quite substantially areas of planting. So what we want to do is ensure that plantings are divided up notionally into 30 metre by 30 metre areas, and the 20 individuals of a particular species are planted within that area so that they're close enough to pollinate. Because if they can't pollinate, what's the point in doing it? So that will do a great deal for the planting. It will ultimately help the remnants, and I'm hopeful that we can expand the remnant areas by adjacent plantings of the genetically diverse planting stock, and to help the problems in particularly the smaller remnants. It will help the gene flow across the landscape, which is critical to the long-term survival of this rainforest. So this also, if this model can be applied to other heavily degraded ecosystems, it's transferable, it's innovative, and it applies the best cutting-edge science we've got to enhance the restoration outcomes that we are achieving. Finally, this is the first way I can see that we can overcome the impacts of fragmentation and isolation, which in an area like this where 99% of the original forest has been cleared are critically important. So I think this is the key to saving our lowland subtropical rainforest from extinction. Thank you.